Hi, Alex. Hi. Sorry, I was running a little bit late there. I had to nip into the house quickly. How are you? How did your test go, by the way? Hi, Alex. Ooh, no, I don't want that to happen. Don't want to listen to myself whilst I'm also speaking. Oh, I'm glad. You seem to be on a roll right now, which is really good. Oh, yeah, it takes us teachers a long time when there's a whole class set to mark. This is the thing, you just got to be patient, unfortunately, which can be frustrating. What shall I do tonight? What topic area? Oh my gosh, did you see actually on that? Oh no, I want that to be up there, don't I? Put you up there. And then I want to. I'm doing the kidneys right now. It's so interesting, but there's so much to do. Yeah, there is, and respiration, these kidneys. Okay, yep, give me 1 second. I just want to show you. Gonna put it on here. I am starting to put some of the year 12 content up on my website. So I'm putting my year 12 content up so you can actually go and read my little notes and stuff. Um, Although that's not looking very good the way it's in right now, because of the size of it. I need to sort that out. But yeah, it's got lots of notes all about magnification. I'm going to add in eukaryotic cells. That's what I've been doing. But I'm just going to put some year 12 content there. Obviously, you've still got my workbooks and my resources. I was just watching your live recording. Like, so. Oh, thank you. I'm glad you're watching my live recording. That's really good. Right, let's look at the kidney structure then. Since you are the first one to jump on in here, we will look at the kidney and go from there. Right, where am I? What am I doing? Here we go. Which part of the kidney are you struggling with the most? This time. You just started got onto the structure, talked about pressures. Okay. So you were doing the glomerulus then, and you're looking at what happens with the hydrostatic pressure. Okay. Let's look at that then. It's greater in the cold part. I forgot the name. The glomerulus. Yeah, right. Let's start. So you obviously know that the kidneys are positioned, they're actually positioned quite high up in your body. So people tend to say, oh, I've got really bad backache, and actually it's pains in their kidneys because the kidneys are sitting quite high up. Now, the kidneys obviously are going to need to be supplied. Second, by, if I move this down here, here we go. Um, Blood which is oxygenated. So you've got your renal artery, which is going to lead into both of your kidneys. And you also have a renal vein, which is going to lead out of the kidneys. So if you think about module 3, and you think about your different blood vessels, and you know that your blood vessels, we have a closed circulatory system. So therefore, our blood is contained within our blood vessels. Our arteries are going to be taking blood into our organs, and our veins are going to be taking blood away from our organs. Now, that's the opposite from when you're looking at the heart. So obviously, arteries take blood away from the heart, and veins bring blood into the heart. But if we're looking at the other organs within the body, it's the opposite way around. So the artery is going to be feeding the blood into the kidneys, and the veins will be bringing the deoxygenated blood away. Hi, I'm very good, thank you. I am teaching about the kidneys is what I'm doing tonight. Now, the connection between the kidney down to the bladder is obviously your ureter, and this is obviously your bladder, and your bladder will lead out of the body where you'll release the nitrogenous waste through the urethra. So when we talk about nitrogenous waste, we talk about our urea, which is going to be dissolved in water, and that's what urine is. Urine is the water and the dissolved substances that need to be excreted from the body, such as urea and oh, okay. My phone, actually, I can take that off now because it's 100% charged. My phone's overheating. It needs to cool down. So yeah, so that's the important thing about the kidneys. Now you will potentially have done a dissection. So this is the kidney that my class were dissecting. And when you dissect the kidney, it's really, really cool to do, but at the same time, it's so soft and slippery. It's quite hard to separate it without causing damage. So my students absolutely loved ripping that apart to the point where they ended up with tiny little bits. But whenever you are doing a dissection, you want to make sure you've got the right equipment. And remember, this is a PAG which they could ask you about in the exam. Just because you haven't done it doesn't mean that you are not going to be asked it. So just be aware of that one. Oh, biological molecules. I was just actually doing that with my year 12 student in my group tuition session. And water. It's difficult right now in year 12, but it will actually get easier because when you go on to the year 13 stuff, it becomes so much more complicated. Nice, you guys are all year 12. If you haven't already done so, do check out my channel with all the videos that will be there to help you. They've been there for the last year. And so hopefully you'll find that a lot simpler. Oh, nice. I love it. People who've watched my channel already. I'm actually on there right now. I haven't done dissection yet, but so excited. I'm sure we will. I have to say the dissection is really cool, but it stinks. The smell, once you open it up and that urine smell that you get, it's an interesting scenario. I love doing it, but it does stink for ages afterwards. Right, when you dissect it, so when you separate and you cut through, being really careful, what you'll actually be able to see is you'll be able to see 3 parts to the kidney. Yeah, it is worse than the heart because it's so much more softer and friable, and the smell, because obviously it's a place where you've been filtering out that nitrogenous waste. And so because you've been filtering out that nitrogenous waste, it has a smell to it. So yeah, it's rough. Especially if it's been frozen and then defrosted. It seems to have a stronger smell that way. Okay, so the key thing we're looking at that you're going to be expected to label is the first bit on the outside, which is the cortex, and then the medulla, which is the middle region here. And you can see that it's a much lighter color. So there's a much darker color around the outside, a much lighter color inside at the medulla. And then you have the pelvis, this section here, which leads down to that ureter. So it is important whenever you're doing this that you note what they will be connecting to. So this is obviously my ureter, which will lead down. And then I have my blood vessels. Now, they won't expect you to label those blood vessels, but they will potentially expect it if they show you the blood is going in and the blood is coming out, or by looking at the lumen size. So you were talking earlier, Alex, about the fact that your teacher has been going on about pressure. So that's another thing to think about. We're talking about blood vessels here. So if we're talking about blood vessels, we need to consider that lumen size. Yeah, they've done loads. So the 2023 and 2022 had loads of questions on the kidneys. So that's a good one for you to go and check if you want to do questions related to it. The other thing they do sometimes make you do, see this is where it doesn't match up put things, is ask you to draw the diagrams. They did ask it in one of the years where you had to actually draw the kidney that would have been dissected. So they do just want you to draw the bits and label them. So making sure your label lines touch, etc. And the other thing to note as well, and I did my diagram on purpose really badly, is that you need to have continuous lines. Your diagrams also need to look like what they do in the picture that they've provided with you. So many students try and draw these like perfect diagrams that they've memorized and drawn time and time again from a textbook, but that's not going to help you to be able to get the marking points for the exam. So it's important to draw what you see, not what you think you should see. Okay, so if we then look at, so you can quite clearly see here, I've labeled with a dotted line, the part which is the cortex above and the medulla below. So going back here, remember my cortex is on the outside, my medulla is underneath the inside. So when we're looking at a kidney nephron, we need to understand that parts of it are in the cortex and other parts of it are in the medulla. And that is important when we're looking at the lip of Henle, because the lip of Henle is the part that goes into the medulla. So you should know labels such as the glomerulus, the Bowman's capsule, the proximal convoluted tubule, the lip of Henle, your distal convoluted tubule, your collecting duct. So they're quite standard names that you need to know, and you should note their location. So you can quite clearly see everything is up here in the cortex, except for the lip of Henle and the collecting duct. What's the lip of Henle? I saw it as a topic, but oh, you haven't started it yet. So the lip of Henle is the region where the water gets reabsorbed back into the body for it to be collected for going back into the collecting duct. And I'll go through what's happening there. But the key thing to note is that you're going to push all the substances out from the glomerulus into the Bowman's capsule. So you're going to do a process called ultrafiltration. And then what's going to happen is that is going to flow through the kidney nephron. And as it flows through the kidney nephron, it always follows the same path. And the reason it follows the same path is because certain areas are going to reabsorb some substances back into the bloodstream. And so we need to talk about what those substances are and how they get reabsorbed. So let me just take this. So you can see it's flowing all the way through. So what we're going to start with is the glomerulus and the Bowman's capsule. No, not just water and urea, salts, yes, but other substances too will need to go. Amino acids, yes. Anything else? Proteins. Ooh, proteins shouldn't really go through unless they are very small proteins. So proteins being located and found in your urine is actually an indication that something is wrong with your kidneys. Hi. Erythrocytes, no, erythrocytes shouldn't get through either because erythrocytes are quite big. So if you're getting blood in your urine, that's a bad thing. Again, that's an indicator that there's something wrong. Ah, Gina, how you doing? Oh, you got a bio test on Monday? Oh, unlucky. But look at it as a good thing because it's going to test what you actually know. Well, this is the interesting thing, and this is what students do, and it's what teachers do. We give you this list of information, and we expect you to remember that. But what can tend to happen is you just literally give this list of everything you know, and you blurt things out that are incorrect. Yes, minerals would be there. If you look at this diagram here, what you can clearly see is a clear section that looks a bit like an ear. I like to think of it that it looks a bit like an ear. This is showing you your Bowman's capsule. And the bit in between, this bit inside, is the glomerulus. And the glomerulus is the blood vessels. And don't say you hate Module 5. Module 5 is actually really good, but there's so much to talk about, basically. You can see the connective tissues, right? Well, it's an interesting part with this one. The staining is actually staining things like the proximal convoluted tubule, the distal convoluted tubule, because they will expect you to understand that your blood vessels run all the way around, and they expect you to note the structures. Now, remember, for us to get a slice like this, what we are ending up with is we're a very thin slice, which has then been stained, differential staining, so we can distinguish different parts within the cell. So what we're actually looking at in here is like a open tube. So the Bowman's capsule, they've sliced it through, and what they're showing you is that region there. So at the Bowman's capsule, what actually happens, one second, I'm just going to move this down here so you can see, is a process called ultrafiltration. Yes, that's a cross section that you can see. That is a cross section. I'll do my own independent version. It's fine. I'm doing okay though. Chidima, do make sure you're checking out the videos on my channel to help you for your test. So what does it actually look like? So what you've got here is my blood vessel, which is going to be pushing blood down into this ball of capillary, and that's what's known as the glomerulus. So we have an afferent arteriole, an arteriole that leads to the glomerulus, which is this ball of capillary. So I've got this afferent arteriole, which is the bit before the glomerulus, and then what's going to happen is the blood is going to be pushed down from the afferent arteriole, and because it's being pushed down at a high pressure, that is going to force the blood down into the glomerulus. So we're ending up with what's known as this high hydrostatic pressure, and that high hydrostatic pressure is going to force the substances, small substances, your water, salts, mineral ions, urea, glucose, amino acids, that list that you were asked to put down, it's going to force them out into the Bowman's capsule to become part of the nephric filtrate. Increase reabsorption. So the podocytes are not for reabsorption. The podocytes are there so that they can actually prevent and help with the filtering process. So when you say the folding with the pods, with the basement membrane, yeah, so what you've got here, talking about that, is here is my glomerulus. So obviously, because we've got glomerulus, we've got our endothelial cells. What we then have is that this high hydrostatic pressure is going to force the small substances, like the formation of tissue fluid. Tissue fluid happens at the tissues. Ultrafiltration occurs at the glomerulus. So basically, we've got high hydrostatic pressure. The hydrostatic pressure is greater than the oncotic pressure. So it's going to force everything, all these substances that are small enough, it's going to force them through between these gaps, these fenestrations between the endothelial cells. Now, Maeve, you've just asked a question about what are podocytes. Well, you should know that there's a basement membrane here that's going to prevent large molecules like your red blood cells, your erythrocytes, like your neutrophils, your plasma proteins. What you also have is you have these podocytes. So this is a diagram that shows you the podocyte. A podocyte is a differentiated cell. So absolutely right, Alex, it's a specialized cell. It's differentiated. And the fact that it's differentiated means that it has got a specific shape. It's got these pods, these extensions, large surface area. And what they kind of do is they wrap themselves around and they interconnect like this. And so what they're basically doing is they are creating another barrier. So the substances that can get through, if they get through the basement membrane, only the substances that are small enough to get through these gaps are going to be able to get through these gaps. So if I were to, I think maybe what they're saying is if I were to increase the gaps between my fingers, larger molecules will be able to squeeze through. So the podocytes are a filtering, a barrier to actually stop the substances from getting into the Bowman's capsule. So they're actually an additional filtering system to try and prevent anything apart from those small molecules going through. So anything that's large should not be getting through. And if there is large substances getting through into the nephric tubule, that will end up with a problem. The kidneys are not functioning properly. So larger fenestrations, again, would be a bad thing. If you've got larger fenestrations, larger gaps, more substances would get through. So the fenestrations should be small to let the small molecules through, but not let anything large through. So the only time the fenestrations get larger is when there has been, for example, an infection and you want more blood flow to an area. Yeah, exactly. You're letting not just proteins, you're letting other things in, and that's not good. So I'm actually on my other channel at the moment, actually displaying the stuff that I'm talking about here. So if you are joining me on TikTok, I am displaying pictures that I'm talking about and answering the questions there. I am jumping between the two. So yeah, so the podocyte is a differentiated cell. So this is where they could actually bring in knowledge from module 2 and assess your knowledge on module 2 to do with the fact that these podocytes are differentiated cells. So because they're differentiated cells, they've got specific shapes, they've increased the surface area, therefore creating that barrier between the two. Oh, no, don't want to do that. Right, so now let's actually look at what happens to the substances. So basically, we're going to squeeze all the substances out of the Bowman's capsule. No, I'm not. I'm going to squeeze the substances out of the glomerulus into the Bowman's capsule. But I have a problem. Always a synoptic link. Always. That's what I do. I have a problem. If I've squeezed everything out of the glomerulus into the Bowman's capsule, I've squeezed out glucose. And if you've done respiration, Chidama, I'm guessing you're doing your test on respiration. If you have already done respiration as a topic area, or if you've already done the hormonal system, you should know that your blood sugar levels need to be maintained at a specific level. So therefore, you don't want to release glucose in your urine, and you shouldn't be releasing glucose in your urine because that is what you're going to use as a respiratory substrate in respiration to break down and therefore use in the chemical reactions so that you can produce ATP in oxidative phosphorylation. So it is really important that you note that if I've squeezed out all my glucose, I need to reabsorb that back in. And this is where your proximal convoluted tubule comes in. So if we know that the blood is going to come into the glomerulus at high pressure, we know the small molecules are going to be squeezed out into the Bowman's capsule. We know that those small molecules are then going to keep going and flowing through the kidney nephron. When they reach the proximal convoluted tubule, this is where we need to selectively reabsorb the substances back in to the bloodstream that we need. So they give you pictures that look like this in your exam, and they expect you to be able to use this electron micrograph. Hold on one second, let's take a step back, module 2 here. They expect you to use these electron micrographs to potentially say that you are looking at a transmission electron micrograph. How do I know that? Well, I know that because it's 2D, not 3D. So this is something I want to potentially make sure that I'm happy with. It's using a TEM because it's a 2D image that I've got. Also, I can see organelles within the cell. So therefore, now don't go down the colored one, Alex. That would be something I would say don't use the structure of it doesn't have any color, because nowadays with the advancement of science and the advancement of computers, etc., we could add color to this picture. So don't use it's not colored. In one of the exams, they actually disallowed that as a marking point. I don't need to apologize. Remember, my aim in these sessions and these videos are for anyone who's watching them to gain the skills that will help them for moving forward in the exam. But one of the things to really know is sometimes you have to look at the detail. So if you're looking at the detail, we can see drawing around the outside of the proximal convoluted tubule. And then we have this like really, if I go back a second, this inner circle, which is not present on this one. So this is the proximal convoluted tubule, and this is the distal convoluted tubule. We can see a difference in the lumen size, and we can also see that this one has what's known as a brush border, where we have an increased surface area. Which allows for more places for reabsorption to occur. So we've got an increased surface area. We've got a smaller lumen compared to this one. So these are things that you might want to be identifying if they ask a question on that. With all the tubules. What do you mean with all the tubule? The tubule, when you're talking about the tubule, do you mean the proximal convoluted tubule and then the distal convoluted tubule? Do you have to memorize them? Yes, you need to know which way it goes. So your PCT comes before, proximal before, DCT, distal after. Proximal convoluted tubule before the loop of Henle, distal convoluted tubule after the loop of Henle. Just because our first lesson was today, so I haven't had time to memorize them. Yeah, but remember, it's not just about the memorization of these words. That's one of the things I would say to you. It's about understanding the topic area and what's actually happening. Of course, of course. It is important because there's so many students who they memorize the content, if I can use my words tonight. They memorize the content and then they come to the exam and they're like, why do I not get it? Why am I not getting the marking points? And the reason they're not getting the marking points is because they haven't actually understood what it is that the exam is trying to assess and what the content is showing them. So what I would say is it's really important that you understand what we're looking at. And that's why I keep saying go back to module 2. So you've got your proximal convoluted tubule. So obviously there is going to be cells there because we're talking about tissue tissues that make up the organ. So the proximal convoluted tubule is made up of proximal convoluted tubule cells. Well, cells we know have a phospholipid bilayer. Cells we know have got organelles inside. So one of the things that we need to understand about the proximal convoluted tubule is that it is a site of selective reabsorption. Well, what does that mean? It doesn't reabsorb everything. It doesn't allow everything to get through. It only reabsorbs the stuff that needs to be moved back into the bloodstream. And so for you to understand selective reabsorption, we actually have to take a step back and understand the membrane structure and the different substances, i.e. the proteins in the membrane. So we have these microvilli, and those microvilli are going to increase the surface area. If there's more surface area, there's going to be more proteins, therefore allowing for more reabsorption to occur of these larger molecules. Thank you for the likes, by the way. Sorry, I am trying to do both at the same time, so that I can give you the content here, but also show it to you here. Now, the important thing for you to understand with anything to do with biological membranes is to understand how molecules can transfer across that phospholipid bilayer. So you know that your phospholipids, how often do I do these lives? Every Wednesday from 9 o'clock till 10 o'clock on here and on my other channel. So the phospholipid bilayer, we've got our phospholipids, we've got our phosphate heads on the outside, which are hydrophilic. We have our hydrophobic tails on the inside, creating a layer. We therefore have a hydrophobic region where substances cannot move across if they are polar or charged or too large. So therefore, for something to move into or out of a cell, it must have the right channels to allow for that to happen. So these cells have differentiated to have the right channels in the right places for the movement of substances, whichever direction that they need to go. So if you understand that, so the hydrophilic heads and the hydrophobic tails, absolutely. What you have to know is actually it links to the active transport of sodium and potassium. So you'll learn about the neuronal communication with the sodium potassium pump. You'll learn about the release of insulin, which involves the sodium potassium pump. Well, also the selective reabsorption that's going to happen at the proximal convoluted tubule is going to be where we're going to be pumping, actively pumping against a concentration gradient, sodium and potassium. No, module 3, you're talking about module 2.1. What says 2.1.6, not 2.1.3. Module 3 is to do with blood and exchange systems, transport and exchange systems, whereas the movement of substances across is, I want to say 2.1 point, hold on, let me think this one through, 2.1.4 or 2.1.5. Yeah, one of those two. Okay, so back to this. What have we got? Well, our sodium potassium pump, but I could relay active transport with plant stuff. Yeah, you can relate active transport with plant stuff if you do it in the right way. This is the human body, yeah. Plants don't produce urine, thankfully. Back to this. We've got our sodium potassium pump. We've got that our sodium is actually going to be pumped out against the concentration gradient. So therefore, that's telling us that there would be a low sodium concentration inside and a higher sodium concentration outside because we're needing to pump it. So if we're needing to pump it, We need to go against the concentration gradient. We therefore need ATP because we need that to provide the necessary energy to pump it against its concentration gradient. So this is where your module 2 should now start to make sense, but actually in context, because we're talking about it, of a job it needs to do, resulting in the selective reabsorption of these molecules. So we're going to pump our sodium out, creating a low sodium inside. And therefore, what's going to happen is, because we've pumped our sodium out, we've got a low concentration inside, sodium is going to flow down its concentration gradient, and it's going to come in through a co-transporter. So because it's coming in through a co-transporter, co-transporter, it's going to bring along glucose with it. So the co-transportation that occurs is the sodium and the glucose together moving in to the proximal convoluted tubule cell, because the sodium is going down its concentration gradient. Now, Alex, when you were talking about module 3 and talking about the movement of substances in plants, the co-transportation is how the companion cell brings in the sucrose into the companion cell, because it pumps hydrogen ions out. The hydrogen ions come back in, and as they come in, the glucose, no, it doesn't, the sucrose comes in with the hydrogen ions, and that's how the loading into the companion cell so that it can then move through into the sieve tube element occurs. So there is a link there, but you just have to know where that link is. Does that make sense? I think TikTok doesn't like me. It's not really pushing my stuff anymore. That's fine. Good. Okay. So the other thing to know is, once the glucose has come in with the sodium down the gradient, because we've gone down the concentration gradient for the sodium, the glucose comes in with it. The glucose will then move through a specific channel, a GLUT channel, which is in the membrane to allow it to be reabsorbed back into the blood. So that differentiation of these proximal convoluted tubule cells ensures that the glucose is being brought in and that the glucose is moving in the right direction. So it's about these proteins that are specific for the role they need to play. Oh, and potassium leaks out as well, by the way. That's the other thing. So if we're pumping the potassium in, because we're pumping the sodium out, the potassium can also leave through these channels, which are called the leakage channels. And that ensures that the cell doesn't end up lowering its water potential too much. So they're able to leak out, which is really good. Oh, I've come to the end of those slides. Does that make sense? Has that helped with the proximal convoluted tubule? And you said you haven't actually covered the loop of Henle yet. So I'm assuming because you haven't covered the loop of Henle yet, you don't want me to go into that detail. There we go, perfect. Have you got any questions about that? Just that away. What am I doing? What am I doing? What I'm doing? Doing excretion. I have to think this one through. Excretion, excretion, excretion. This one. So if we were to look at, for example, 2023's paper, 2023 gave a picture that looked like this. The thing is, what you've got to remember is topic areas. If you want to get ahead and pre-learn, then you need to just jump on my channel and watch the videos that are related to topic areas that you are watching. They are there ready for you. So yeah, definitely go and check those ones out. Because that will help you either to recap what you're doing or to get ahead. Oh, do you? Why are you not asking questions in the videos then? It's a funny one. Homework is there to help you recap and go back over, but sometimes as teachers we do give too much. I don't apologize at all. You don't need to apologize for not watching my videos. Watch them every day, every second of the day. They're there for you when you have time. This is the thing. It's finding the right balance between you also having a life. That's what you've got to remember, that you also need to have a life as well. It's not just about you doing what is here. I do these sessions because I want it to be that I'm available and I can offer a little bit of help for some students who can't afford tutoring. Because obviously I have my private clients who come and see me one-to-one. What's the pelvis? Which one? Oh, thank you. I'm glad I'm helpful. I do try. Give me 1 second. I just need to do my here. So V is the pelvis. Good. What else have we got? It's not a problem. Yeah. I have to, I'm also needing to edit my video from my group tuition so I can send that out to the students who attend that. U is the cortex, good. So therefore, what is T? Because such an itchy nose. And then if we look at another question, T is the medulla. Yep, absolutely right. So T is the medulla. And then if I show you the next question, again, this is 2023's question. So you can quite clearly see that 2023 had a lot on the kidney. So this is the next one. And it wants you to use those previous letters. So you've been given a cross section of the kidney. You're being told that oh, that's plants actually are really cool once you get them. You've been told that you want to use the letters T, U, or V from the drawing. So from this one above, you want to use T, U, or V to identify the part of the kidney that used to make the cross section in the photomicrograph, and then give a reason for your identification. So what was it about this that actually allowed you to work it out? So if you can see, you've got your T, you said was the medulla, U was the cortex, and V was the pelvis. Identifying from there, what is it? Is it T, U, or V that is actually allowing you to identify what you're looking at? Okay, so if we go back up here then, so this is looking at a picture. Da, 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 da. Where's it gone? Where's it gone? Where's it gone? I went too far. So here we have here. This is the cross section, isn't it? You can see that cross section. You can see the clear region here, C section here. So what are we looking at? We're looking at the glomerulus and the Bowman's capsule. Yeah. And where is that located? That's located at the top region. So if we go back to down to the question then. Oh, went too far. What can we see here? We can see our Bowman's capsule. There we go. So therefore, because we can see our Bowman's capsule, we know we're looking at the cortex, and therefore we must be looking at, because we rightly identified you as the cortex. Why? Because we can see the glomerulus and the Bowman's capsule. So they're the types of questions where it's assessing your knowledge of you being able to apply it to the question. So they expect you to have seen those cross sections. They expect you to know about them. And so they are wanting to ensure that you've got that ability to apply it to that question. You are very welcome, Alex. You are very welcome. I hope that's been helpful for you. And that's the thing. Once you have it, once you know the topic area, once you have a part of a module, it's then just about doing practice questions on it, because you think you know it, but actually maybe in a week's time, you're like, oh, what was that again? And it's about going back and rewatching it. The other thing to note as well about this is they've also given us a magnification down there. So that means that if they were to use this picture again, they could give you a question which expects you to use your magnification equation. Oh, absolutely. Spaced repetition is so key to make sure you understand the topic area. They could ask about the image size. So they could give a question where they say, right, what is the size of the glomerulus? So measure from A to B. Or they could give you a scale bar and then expect you to use that. If that scale bar is 5 micrometers, then can you then tell how to tell is not what I'm trying to say. You can tell I'm getting tired now. Can you describe how you would use that scale bar to figure out the actual size of the glomerulus, for example? So there are lots of different ways that they can ask questions. It depends what they want to ask. They could make it all about microscopy, absolutely. They could ask about the eyepiece graticule, absolutely. It just depends what they want to do. It depends what they haven't asked in a while. It depends where they've gone, because that is obviously module one. Paper 1 is module 1, 2, 5, and 3. Whereas module, look at me mixing everything back to front right now. And paper 2 is modules 1, 2, 4, and 6. So they could ask whatever they want, or if it was paper 3, it could be all of the modules. So it really just depends. And although you say, oh, typical OCR, but that's the whole point in OCR and the way that they ask the questions. AQA have those short questions that they ask you, and then they have the long write your answer 25 marker on paper 3, linking different topics together. Or with OCR, they're different because they're actually asking you short questions. The longest you'll get is a 6 marker, but it's very much still got to assess the whole topic area and find those links that come together. So it's just a different way of assessing with the exam boards. But it is now getting time for my bedtime, so I am going to go. Good luck with the revising and the topic area, and good luck when you get your test result back. I hope Chidima is doing all right with her revision for her test on Monday. Bye. Have a good evening. Thank you to those of you who have watched this. If you are watching it, please do like and share so that I can keep making more videos, keep doing more and more of these. Because the more people who find my resources, the more people I can help. So please do like, share, and I look forward to working with you guys again next week. So see you next Wednesday.